Well, thank you so much. It's really a wonderful honor to be here. This is my first trip to Egypt, actually. So I don't know why I waited so long, but it's, the hospitality's been wonderful, and the scenery is also lovely. Red Sea is great. So I'm going to talk a little bit about decidualization. You heard a very nice presentation about the practical application of progesterone in early pregnancy and actually for the prevention of preterm birth from Professor Mona. I'm going to talk a little bit about uterine factor infertility and the role of the decidua. Now, as it turns out for all of us, really probably the most important day of our lives was the day that we implanted as embryos. And as you're aware, mammalian reproduction isn't very efficient, and it's particularly not so actually in women. So the iceberg is to really kind of demonstrate that the majority of pregnancies that are conceived are actually lost spontaneously. This process shown down at the bottom where the embryo makes contact with the endometrial surface epithelium and begins the process of implantation is one that's really quite difficult to achieve. So in normal spontaneous pregnancy, only about 30% of those conceived pregnancies actually give rise to live births. And in vitro fertilization, that process is even less effective, with sort of less than 25% of embryos implanting. Probably in part because women and couples undergoing IVF practice are hypofertile in the first place. But this process of implantation is really critical to the establishment of a healthy pregnancy, and that's been an area of interest in my laboratory for several years. We've reviewed this already. You're all familiar with the endocrinology and the development of the endometrium here in what we refer to as the window of implantation, a fairly short period of time, lasting probably 72 to 96 hours in total, when a healthy embryo actually has the ability to implant in the uterus. And trying to understand how this process works has really been a major focus in reproductive medicine for many years. Now, we've learned quite a bit over the years about the mechanisms of action of the steroid hormones. This is just to demonstrate that for estradiol action, there are receptors within the cell, which become nuclear receptors that activate gene transcription. There are also receptors at the cell surface that are important for various signaling processes. And this gives rise, estrogen really sort of drives gene activation and particularly cell division, proliferation in the endometrium that you're all familiar with, that occurs during the proliferative phase of the cycle. In the secretory phase of the cycle, progesterone is secreted from the corpus luteum, as we've heard earlier this morning. Its action or function within the cells is very, very similar to that of estradiol. There are nuclear progesterone receptors, two kind of versions of those. There are membrane progesterone receptors that are also really important in mediating the effects that predominantly have to do with gene activation and cellular differentiation. So estradiol makes the cells proliferate, it prepares the cells to respond to progesterone, and then the superimposition of progesterone in the luteal phase of the cycle allows the cells to differentiate and be prepared for embryonic implantation. Now, clinically, we have some measures that we can follow to look at the development of the endometrium. Ultrasound, and you'll all be quite familiar with the kind of the trilaminar proliferative phase ultrasound picture that we see. And in the secretory phase, the endometrium becomes quite edematous, bright white sort of appearance on ultrasound. And the thickness of the endometrium tends to correlate quite well with pregnancy outcomes, but there's really no specific cutoff. So here below I'm kind of showing that the clinical pregnancy rate has a tendency to increase as the thickness of the endometrium increases, but you can't really identify a single point where this is a healthy endometrium, this is not a healthy endometrium. It's really a linear sort of spectrum. And the opposite is there's kind of a negative correlation between endometrial thickness and the risk for spontaneous abortion. Again, it makes sense to us that the endometrial thickness is really important, but we can't identify a single cutoff point that we can use clinically. So we've attempted to use other kinds of approaches to understand endometrial function. And one of the classic ones is demonstrated here, the old Noyes criteria, published several times in the 1950s, of the proliferative phase, the secretory phase, the kind of changes that occur histologically. But as we know from the work of Christos Koudaferis in a publication in the New England Journal several years ago now, that this histology, classic endometrial histology, isn't very predictive of pregnancy outcome. So we know that the endometrium is important, but we don't really have very good handles on understanding that. In recent years, the work of Carlos Simon and others have really demonstrated and taken the approach, if we could understand the molecular development of endometrium, we might be able to identify more precisely the time that the endometrium is receptive or not so receptive to implanting embryos. And his work has identified these 238 different gene profiles from biopsies taken from patients with pre-receptive, receptive, or post-receptive endometrium by looking at a whole array of different genes that he's been interested in his laboratory. Has actually found a signature, essentially a fingerprint of the endometrium that's receptive. And this ERA, endometrial receptivity assay, is one that he's now made commercially available. And there are other kind of versions of this that are being developed to try to help us understand, if we can identify when the endometrium is receptive and when it's not. So we have ultrasound approaches that aren't very precise. Hysteroscopically, we can look at the endometrium and get a sense as to whether it's healthy or not, but it's very difficult to precisely define the receptivity of that. We believe that the old Noyes criteria aren't as predictive of endometrial receptivity as we thought, and we're hopeful that some of these new molecular kinds of analyses might lead to better tests that we can use clinically to identify. And I know that some of you are using this kind of testing already. It's being done in sort of artificial cycles currently because it's most useful in that setting. So I'm going to go into a little bit about some of the interests in my own laboratory. We haven't studied 238 different genes, but we've studied a number of these, and so these are some of my favorite genes, some of which actually correspond with the ones that Professor Simon has studied. And here's a whole list of these kind of crazy gene names and proteins. It's not really important that you understand any of those. I'm going to use one just as an example of the kinds of approaches we're trying to use to see if we can understand how one might sort of manipulate and change, sort of promote the development of the endometrium to improve pregnancy success rates at the time of implantation. So I'll talk about a protein called connexin 43. Now, connexin 43 is a gap junction protein that actually allows two adjacent cells to communicate their intracellular contents. In English, we kind of would call that spot welding. If you take two pieces of metal and sort of fuse them together with a weld, that's a little bit of what's shown here in the pictures with the arrows. And these proteins actually have 6 subunits and sort of form a pore that actually allows two cells next to each other to exchange some of their cytoplasmic material, particularly small ions and small proteins across that. What we learned in the mouse is that if you knock out this particular gene, connexin 43, in the mouse uterus, if you knock it out in the entire mouse, the embryonic mice die because their hearts don't develop. Connexin 43 is also very important for cardiac development. But if you just knock it out in the uterus of female mice, they have a very poor implantation rate. So in wild-type mice, WT, the embryo can make contact with the endometrium. But in these mice that are missing connexin 43 from their uterus, the embryos don't attach very well. Very few will actually have pups and deliver pregnancies. So we knew from a mouse model that this was important for implantation. In women, one of the situations we studied that's an area of interest in my own laboratory is an endometriosis. And what we identified is that this connexin 43 protein, which, as you can see on the left here in normal women, is predominantly in the endometrial stroma. The epithelium that overlies that has very little of the brown staining. Connexin 43 is mostly in the stroma of normal women. In women with endometriosis, the levels are significantly lower, probably less than half that protein concentration in endometriosis. So we thought if this is similar to the mouse, and in the mouse when the levels of connexin 43 are very low, embryos don't implant very well, maybe this is in part the problem of infertility associated with women with endometriosis, that in fact there's a failure of implantation in embryonic development, early pregnancy kind of loss as a result. We can take biopsies from the endometrium the way we do for clinical histological confirmation or purposes, or the way you would do for the endometrial receptivity assay if you were sending those samples to one of the big laboratories that does that kind of study. But in our own laboratory, we can grow epithelial and stromal cells from the endometrium, from women with and without diseases that we're interested in. And as I mentioned, because connexin 43 is in the stroma, I will kind of focus on the stromal cell cultures that we've done. And these cells can actually be treated in the laboratory with estradiol and progesterone, trying to recapitulate the sort of hormonal environment that's typically seen in the luteal phase of the cycle for 7 to 14 days. And you can see on the top the cells, which start out looking very much like stromal fibroblasts, when they're exposed to the steroid hormones for 7 days or 14 days, actually become quite plump and epithelial-like. And that's part of the decidual response that actually allows those cells to be receptive to embryonic development. And if we remove the hormones from the culture, the cells actually go back and revert. So they're very dynamic. They start out as kind of thin little cells, become more epithelial-like as they differentiate and then can return. And during that process, a series of products from the cells increases and decreases. Prolactin is one of the proteins that goes up. Vascular endothelial growth factor and angiogenic factor that's important for vascularization of the endometrium goes up and down. And interleukin 11, another sort of cytokine that's important for implantation changes. And during all this time, connexin 43 in the brown, you can see that that protein goes up and also goes down again as hormones are added and then removed from the situation, from the culture. So this really kind of reproduces the kinds of things that goes on in the human uterus during this differentiation process. Now, some of this is a little maybe overly scientific, but I wanted to point out the idea that maybe we can take other approaches to try to increase the production of this protein in women who have progesterone resistance or who have other kinds of endocrine problems. And this demonstrates really that we can use genetic approaches to overexpress connexin 43. When we overexpress connexin 43, a series of different proteins goes up and down, but one that's quite dramatic is this interleukin 11. So without adding hormones to the cells, we can actually genetically overexpress connexin 43 and get some of those hormonal changes to occur as if the cells were exposed to hormones. In the endometrium, we believe that the crosstalk between different types of cells is really very important and critical. And perhaps one of the most important is the inflammatory response that occurs in the endometrium, where macrophages are attracted into the endometrial tissue. They secrete various cytokines interleukin-1 beta being one of the most potent of these inflammatory cytokines. And in turn, the endometrial cells can influence the recruitment of immune cells. So shown up here is some endometrium. I think that's, well, it is, but it doesn't show it very well. The green cells are macrophages, CD68, macrophages coming into the endometrium, and they release interleukin-1 beta that causes the endometrial stromal cells to then become inflamed. And the cytokines, prostaglandins, reduce the connexin 43, and that leads to a decrease in receptivity. So this demonstrates that these cytokine proteins, like interleukin-1 beta, can cause quite a dramatic reduction in the connexin 43 at the top line. And you can see also in the cell cultures themselves, the little green dots, the green spots are actually connexin 43 proteins. When interleukin-1 beta is added to the cultures, this inflammatory environment actually causes that protein to be dramatically reduced. So we believe that part of the problem with endometriosis and possibly other inflammatory diseases associated with poor pregnancy outcome is the ability of these inflammatory cytokines to reduce the production of this very important protein for receptivity, embryonic receptivity. So to conclude that, we believe that the preparation of the endometrium for embryonic implantation is really critical for pregnancy success. We know a lot from our IVF experience about regulation of the ovary and folliculogenesis. We don't understand very much about what happens on the endometrial side of this, and hopefully these will provide some insights. We can use ultrasonography and hysteroscopy as a way of assessing endometrial responses, but they're not very precise, aren't really very accurate, don't allow us a lot of prediction just based on the thickness of the endometrium alone. And we believe that the sort of traditional noise criteria of histology isn't very predictive, but we're hopeful that newer approaches, the ERA and maybe some specific proteins that are present within the endometrium might give us a better idea about when the endometrium is prepared and when it's not. We've examined in my own laboratory this idea of endometrial stromal cell differentiation in women with endometriosis or not, or other forms of infertility, and also in vitro in the cell culture model that we've used to try to understand how that's regulated, might give us some insight into sort of recurrent miscarriage and other implantation failure. And we know that we have the ability now to manipulate the expression of connexin 43. We can reduce it. That doesn't help us clinically very much, but it helps us understand inflammatory conditions that might be important. And ultimately, we would like to find ways to stimulate the production of connexin 43 so that we can potentially augment endometrial function in women who have implantation failure or infertility as a result of inflammatory processes. So I will go ahead and stop at this time, and we'd be happy to answer any questions that you might have. Thanks.